Hello, everyone. Good morning, good afternoon, wherever in the world you are. I'm Azadeh Tafreshiha. I'm the project manager at the Hive here in the Netherlands. And welcome to the second of the OMOP series of webinars we're having. This one all about important topic and somewhat maybe neglected sometimes, data quality in real-world data, specifically when using OMOP. So to get us started, I thought we'll share a poll with you to hear a little bit from you, whether you use data quality tools or not. So we do have two very nice speakers who will answer your questions. First, we'll hear from them for 15 minutes each, and then there is time for Q&A. So please send all your comments, questions in the chat or in the questions part of the platform. I'll make sure to get them, and our speakers will answer them. So first up, from United States, please, Alex, introduce yourself.
Hi, Azadeh. Hi, everyone. My name is Alexandra. I'm so happy to be here today. Looking forward to the discussion that we'll have in just a little bit. I'm a senior product manager at Kythera Labs, where I work with different types of real-world and observational healthcare data to develop data and AI solutions for life science use cases. My background is in implementing digital solutions in clinical trials, and before that in human subjects research. So I generally like to work on problems that involve making sense and scaling up lots of different types of data for research use cases. And I'll hand it over to Liam.
Thanks, Alex. So hi, everyone. My name is Liam Gluck. I'm a data engineer at the Hive, where the large majority of work is specialized in the OHDSI OMOP Common Data Model, as well as the tool suite and the open source tools that are utilized often around this, which I'll be showcasing a bit to you all. My background, at least as a data engineer, I have a background in biomedical sciences as well as computational biology. And I'm also very happy to be here with you all today to hopefully showcase why data quality is not as simple as it may seem, but just as important to keep in mind when going on this journey. Should we get started then?
Yes, please.
So let me. I'm hoping that you can see the screen.
Yep.
Is it gone full screen now?
Yes.
Yeah. Perfect. Okay.
Yes.
So I won't be able to see the cameras or chat, so please let me know if anything. That should hopefully be around. Yeah, you see the title page this time, right?
Yes.
Okay, so hi everyone, thanks again for joining us. Just to reiterate again, our webinar today will be going over the journey of your source healthcare data being transformed and mapped to the OMOP data model and how to effectively utilize data quality checks implemented within this process. So just to give us a little agenda of what's to come, I will be going through how assessing data quality throughout this process, what it can look like, mainly what are the OHDSI tool suite, especially in regards to assessing data quality, the multiple levels that data quality can be checked and implemented in this process. So initially, I'll talk about assessing your source data as well as the DQ tools specifically for your OMOP data. Specifically going into a little bit more depth on the data quality dashboard, Achilles, as well as the CDM onboarding tool. So what do I mean when I say the OHDSI tool suite and the journey that one might go on when typically transforming data from source data to the OMOP database? This is a sort of a visualization we like to use, where on the left, you would have your typical source data starting off, whether you're an organization, a hospital, an institute, or any holder of observational health data that can be transformed. It's good to know that there's quite a plethora of open source free tools available to you that can be utilized that are highly developed and specialized towards mapping different data to OMOP. As you can see here, the white rabbit, the rabbit in the hat, the Usagi and Athena can help you in your process of aligning your needs on how you will map the data. An ETL pipeline tends to be a little bit more of the unique cases among the individuals mapping the data. This might be how you implement it as a group, as an individual, as a company, as well as then the tools utilized once you have your data in the OMOP CDM, such as the DQD and Achilles, and how these can be utilized in order to reiteratively check your data quality. So as I said, this is an example roadmap. It's good to keep in mind that with all these tools I'm showing you here, they are open source, they're well documented. They've been developed specifically with mapping to OMOP in mind, but that is not the only use case that they can be used in. As well, they are maintained by the community, by the OHDSI community, or the Hive ourselves. So, and good to also mention that the webinar will obviously specifically focus a little bit more on the data quality tools in the red, as you can see, DQD, Achilles, as well as CDM reporting. So what do I mean when I say that data quality should occur on multiple levels? And as you can see, you'll have an initial step of your source data towards the ETL to then the OMOP data. And data quality is something that should be implemented on every one of these steps. It's not just about validating your data once it's in the OMOP standard, but also checking your source data as well as the ETL pipeline. You've got your source data, whether that be EHR, registries, secondary or tertiary healthcare. But before you start the conversion process, you have to look at the quality of your source data. You want to know how well is it fit for the ETL? Does it contain errors that are only going to pop up to a much more significant extent later on? These rabbit tools, as we call them, are very useful for the initial exploration of the source data. Whereas for ETLs, this is very often and most commonly tested and interrogated with end-to-end and unit tests. Whereas then once your data is in the OMOP format, there's a suite of tools available to be utilized that can assess your OMOP CDM standards, ranging from automatic checks, combing through every table, to general reports and statistics of the vocabularies utilized. So what exactly do I mean when I say that the rabbits will help you understand your source data? So White Rabbit specifically will summarize and profile the fields of your source data. It will give you informative summaries of each important field, the counts, the uniqueness, and the emptiness. It lets you understand the limitations of each field and the values combined. Rabbit in a Hat, on the other hand, will take the scan report output of the White Rabbit as input and allow you to then manually define mappings from your source to your target tables, often in a clear way that can also be exported to documentation. You can make comments about the logic that you can make sure that your colleagues will see in your sort of collaborative process. And another hugely useful feature is that it exports a very large and automated file that can be utilized as the test framework that you can then base your unit or end-to-end tests off. Good to mention that that's in the context of R, right? Then third-wise, Usagi is a very useful tool for mapping individual values and entries that are not so straightforward that you might see in your source data, right? They might not be represented by standard codes. Perhaps you come from a country where very specific framework or ontologies are used, or you have a lot of free text fields that have variations in their spellings, but you still want to map the fields. So what Usagi will do will incorporate vocabulary files from OHDSI into its string similarity search tool. And this will give you a nice mapping table that you can then use for your ETL process as well, but as well also documentation of issues or questions you might have that you and your colleagues will want to work on together. So now we move on to, once your data has actually been OMOP'd, what are the relevant tools that we can utilize in order to really interrogate that OMOP CDM database? And the three tools I will discuss here are the Data Quality Dashboard, Achilles, and CDM Onboarding. So the DQD tool is an R package. The DQD tool is an R package that has been developed and is maintained by the OHDSI community. It is part of what they call the Hades Analyticals tool suite, even though, yes, it's utilized in this analytical end state, just much more for this iterative data quality checks. I will go a bit through the contents of what really makes up these data quality checks. So these are organized according to something called the Khan framework, which uses a system of contexts and categories to represent strategies for assessing your healthcare data. So within these contexts, specifically within the DQD, it mainly focuses on verification in the sense of It focuses on how your data values that occur there match to expectations with constraints, system assumptions, as well as local knowledge of this data. Whereas then verification focuses specifically on the alignment of data values with respect to relevant external benchmarks. And we'll make this a bit more clear later on. But what's important is to understand the CON framework will combine these contexts with what they refer to as categories and types. So these categories and types represent the different methods to validate your data then. So conformance, for example, conformance checks are for whether your data follows the required format of the OMOP CDM, the structure, the rules that we try to abide to ensures that values are in the correct format and meet the expected standards, right? So do you have duplicate fields in your person ID fields, which need to be unique IDs? Do you have records that are not utilizing standard or valid concepts in the OMOP field, such as the drug concept ID, which relies on you utilizing standard relevant codes? Completeness, on the other hand, checks how much data is present in the data set without considering its accuracy or structure, right? We're talking about whether any data is missing or such. It checks that every person in the CDM also has a relevant observation period table, or that there are no null values in dates where, in theory, nulls are not allowed. And then lastly, plausibility. So this is a check on whether the data makes sense and seems realistic. And this will look at the value patterns, the distributions, the relationships, and between these data points, and ensure that they align with our expectations, right? So is it that your measurement date is occurring after death? Is it that an important procedure only occurs before the patient was admitted to the hospital? All of these things will be checked and alerted to you in a sort of a pass or fail framework. So what do we mean with these context and these checks and these types? So you might have 24 check types, and the combination of this plausibility, conformance, completeness with the verification and validation. But the strength in DQD is that it utilizes these check types to then scale all these up for all the different kind of source fields and concept IDs that need to be checked. And that's what makes this a nice extensible framework, where in the end, if you want to run every single check, you'll be implementing over 4,000 checks. What do the results look like? In essence, this is just a simple output of what the Shiny R tool will output your JSON results as. So this red corner in the box, as you can see here, is a nice bit of R code that's quite easy to implement. You connect it to your database, and then you specifically run it. As you can see here, this nice little tabular output just gives you a really nice scorecard output and summarizes all the checks that have failed or passed and give you an overall percentage. And obviously the aim is to increase this overall pass percentage, but of course it's understanding the nuances of your data, and sometimes this might not always be possible, right? So let's move on to the next tool then, which in this case is Achilles. Now, Achilles stands for Automated Characterization of Health Information at Large-Scale Longitudinal Evidence Systems. And this is a software, again, developed by the RC community that provides, as the name suggests, it characterizes and visualizes your CDM database. It's a well-made R package, and that produces, the main use of it is that it produces reports based on the summary data it generates. As well, it will also have an additional function to help with the use of the Atlas tool from the Odyssey. That's a little bit out of our scope for this webinar for now. So it's just to keep in mind that it consists of two parts. First being the description of the databases and the statistics. It has great features to not only provide important features, but also output for the next tools, such as CDM onboarding, which we will discuss. And it can also be implemented with a nice HTML website base to give a series of interactive summaries and visualizations. But what's good to keep in mind, especially, is that it provides the output for a lot of the figures and analyses done for the next tool, which is the CDM onboarding. Now, this is the third and final tool that I will go through on this list. And as I said before, it takes the output of Achilles, so all these figures and summaries and statistics to make and what CDM onboarding will do will make a rather extensive and cohesive report on all of this quality from your OMOP CDM. This was specifically a tool that was developed within the Darwin EU initiative, created to be run on registered data partners databases, so that the sort of network operations that works with the EMA can have a very good idea about the data quality that's coming through without actually having to touch and deal with these very sensitive databases, right? Dealing with hospitals and patient data. So I'll go a bit more specific into what this package will show you, because unlike the DQD, it's good to acknowledge that the CDM onboarding will not give you a pass or fail, right? This is a tool that summarizes and reports everything to you, and you have to have the little bit of the nuances to go through this and understand what's important to look at. So what I mean here is that the CDM onboarding, on top of interrogating your execution details, it will check what packages was used, the CDM, the vocabulary versions, the date of executions, the release dates of your data to make sure everything lines up nicely. It will show you the record counts of each OMOP table, visualize your date ranges, as well as a period of events. This is an example of what I mean. You have to know what to look for. Are you seeing any very big dips or any very big peaks or low dips in your data? Date ranges are a huge aspect of the CDM onboarding because the OMOP Common Data Model really depends on the utility of your dates and how well they define the events. So they're very important for abiding to the rules of convention, and in the end, the quality and readiness of your data for research. So you'll have to manually check these summary tables to make sure that nothing pops out, that you're not seeing dates from 1850, or you're not seeing measurements only happening in 2030, for example. So as well as the nature of visits are shown, you can check, okay, outpatient visits should be a certain length as compared to inpatients, and all of this relevance, as well as the events and how they occur during the month. Is everything occurring on the first day of the month or midway through the month? And this is normally an indication of the transformation process that's gone through rather than the source data. What's also really important is the vocabulary mappings, as OMOP depends a lot, and its strength depends a lot on the vocabularies used. It's a huge component of the CDM onboarding. It will check your mapping coverage, the granularity of your detail in your drug mappings, as well as both the most commonly mapped as well as the most unmapped tools. So the drug granularity is quite important for networks, for example. You want to make sure that you, as a clinic, a hospital, a company, organization, are actually embedding information in your drug mappings, such as the amount that the patient is getting, the time ranges that they're getting it for. This is what this tool will help you analyze better. Unmapped codes, in essence, in this case, is more so for letting who is ever sort of performing the data quality check to see, okay, are there any low-hanging fruit there, right? Do you have codes that could be mapped that you just don't have mapped right now? Or as well as you can swap this relationship to look at the mapped codes and check, is everything as it should be there? Are we seeing the most common procedures occurring as the most common procedures? And same with the drugs. For example, hospitals, you'll expect to see pain relief medication to be the most common, whereas cancer registries, you're more likely to see chemotherapies as the most commonly utilized drugs. So it's nuances and information about your source domains that's going to be important for this. In the end, I wanted to come back to this sort of view of the multiple levels of data validation and how they occur. It's really good to emphasize that these tools are meant to be used iteratively, right? Especially when I'm talking about the last three, the DQD, Achilles, and CDM onboarding, because this is an iterative process for you as a team of engineers, database managers, data scientists. When you transform your data once to OMOP and you run the DQD, Achilles, and CDM onboarding tools, they'll realistically show you the limitations of the work you've done so far. It's your job then to correct for the work you've done, document it, and rerun these tools again to make sure that you're hitting a higher level of then these data quality metrics, right? It's not just a it's not just a run this one time and everything is golden. Standards alter over time. Vocabularies are edited, they're improved, they're changed, meaning care has to be put in to make sure that everything in your database is also standard and still informative. If you want your data to be ready for research and representative of the data captured in your source, then these data quality tools will be of immense help for you to make sure that your data is research ready for analytics and any use case that might be important to you. Thank you for your attention. I think the next part I'll hand over to Alexandra.
Thank you so much, Liam. This has been a really, really nice introduction, because what we thought we would share with you next is a little bit more of a large-scale OMOP implementation for which Kaithera Labs and the Hive partnered. So we can go to the next slide, Liam.
Yep.
Thank you. I just want to give you a sense in the next 10-15 minutes what we'll cover. I'll start by talking really briefly about the data fragmentation challenge that a lot of us working with various types of real-world data often experience. And then I'll walk you through key elements of the implementation, OMOP implementation use case I just mentioned, where we really worked hard to define and design an OMOP remastering process such that we have the ability to build up with scale and with a lot of precision and auditability into the pipeline. So I'll tell you a little bit more about key technology enablers that we found were really essential for enabling ETL development and some of the standard community tools that Liam overviewed in a cloud-based platform. And then we'll discuss lessons learned for our organizations and next steps in our OMOP journey. So without further ado, we can jump into the problem space. The next slide. Many of us here are real-world data practitioners, so I probably don't need to say much more than just to validate some of the typical pain points that we can experience when trying to do research with data coming in from claims, electronic health records, lab tests and results, registries. It's often very complex data. It comes from different places. And what this means in practice is that trying to integrate those sources for research purposes can be slow, it can be expensive, and sometimes error prone. What we often see in research projects is repeated data wrangling, remapping of data on a per study basis. That is typically what drives costs up and really slows down research, which we don't want to see. That's why at Kythera Labs, we turned to the OMOP format because we wanted to leverage that shared global structure, which is really, really important so that we can speak the same language across organizations and within our own organization as well. But one thing to keep in mind is the OMOP data standard in and of itself would not necessarily help as a standalone data standard to solve for the fragmentation and interoperability challenges. So we think to make the most out of OMOP standard with very large observational data sets, which we'll tell you a little bit more about in a minute, you have to really be very thoughtful with how you set up the mapping process from your sources over to OMOP format. And we really also have a nice opportunity here to bring in modern platform technology tools so that we can build an infrastructure that's highly reproducible and transparent. And we'll focus on that in some of the next slides. So let's take a step back just for a moment. To make OMOP implementations reliable at scale, we do need to make sure that when we're working with very large, complex data sets, and those are coming in from different places, we have the right operational foundation. So this is where I wanted to mention our Wayfinder platform on which we built our OMOP implementation. Wayfinder is built on Databricks. And before we even think about our OMOP formatting, we want to make sure that any data source coming in is findable and accessible. So this is where we leverage some of the Databricks Unity Catalog features because we are working with a searchable index and secure access controls by default. What this means in practice is when you bring an expert or a partner like the Hive's team, you're reducing data duplication and data sprawl. We don't need to exchange copies of data sets. We can provision access safely and securely on a per user basis. And we can also restrict, of course, modifying or changing the source data tables. And then we also make sure whatever sources we're thinking about converting over to OMOP format, that those sources as standalone sources are interoperable and reusable. So a couple key improvements that Kythera Labs implements before OMOP transformations to mention here would be data de-identification and tokenization, and also making sure that when we build certain transformations or improvements, we have that provenance and lineage embedded in our code base in all that we do on Wayfinder, because we want to make sure that the transformations are transparent, reproducible, that we can edit, modify, every step of the journey, from ingesting a source all the way to generating our OMOP data set. So bottom line here is that we have modern platform capabilities that help us make the most out of data sets in a reusable, auditable manner. We can move on to the next slide. So here we just wanted to tell you a little bit about the process from left to right on the slide, moving from raw data all the way to an asset like an OMOP database. So typically for various data sources, claims, EHR, labs, those are some of the sources that we recently transformed to OMOP. You start at the bronze level with simply landing, bringing over the data as is from whatever the source is. At the silver level is where we start to impose certain schema transformations, requirements, basic augmentation of the source. This is where we start to really think hard about structure and consistency. And then at the gold level is what we consider as analysis-ready data. It's updated, it's cleansed, it's standardized. Downstream teams and users can put the data to work for research purposes. And then oftentimes what we see, especially when working with OMOP data, is we want to bring over some of those familiar open source tools, right? So the platinum level, we want to complement the data with both Wayfinder, standard tools. I mentioned to you, for example, the Delta Share protocol. We don't want to be making copies of the data. We want to be smart about how we access data. And same with the data quality dashboard, Achilles, and other tools that we'll mention in just a moment. It is at that layer that those tools become available for users. So the big picture here for me is that by the time you stage and prepare your data for OMOP transformations, you want to make sure you're not just mapping from your raw data sources. You've staged structured, reliable input that you can manage and govern, and then you turn over to the OMOP data set. And this was a very specific design decision that together with the Hive, we applied to this particular implementation. We can go to the next slide and take a look at what that looked like in practice. So we selected 2 EHR databases, one laboratory test and results database, and a large claims vendor database coming from a clearinghouse. So that is pulling from several different sources, all US national data sets. What we did with the EHR and labs data, as well as claims, is we separately harmonized those into consistent internal data models. This is something that we do at Kythera Labs by default. It's a part of our DNA. The reason we do that is because anyone here in the audience would probably relate. If you've touched a raw claims data source, it's very complex. You might see situations like you have a surgical procedure that has occurred, and that procedure has generated 5, 7, or more medical claims per patient. What you want to do is you want to spend some time rationalizing these into one coherent event for that patient. You have an opportunity to adjust for missingness, align the provider, align the point of care. And we think that it's really valuable to do that work in advance before you carry over your input to the OMOP format. So again, here what we are after in this implementation is making sure that we stage the data in a smart way that would allow us to remove basic ambiguity in the data so that we don't persist it into the OMOP schema. And also there's one more thing here, which might be not intuitive to notice from the diagram. When you stage your data in a specific way before mapping over to OMOP, essentially what we're eliminating here is the need to move from each source separately, build your ETL over to OMOP format. We want to be flexible with the sources. We want to be able to turn on and off different sources. We want to be able to augment the OMOP implementation with additional data sources. And so we don't want to have to redo and manage individual pipelines that's not scalable. We want to prepare the data, make sure that meets certain requirements before it goes over to the OMOP format. And here also, it's a really nice opportunity to highlight a unique collaboration on the Kythera Lab side. We are very good at thinking about how to stage, how to cleanse, how to prepare the claims in EHR data so that it's ready for OMOP. And then we bring over the Hive core experts at helping us make sense of the data semantically, as well as to figure out exactly how and what elements to carry over to the OMOP standard. So it's been a really unique, really powerful collaboration in that sense. We can go to the next slide. So here we're starting to highlight just a few basic results. We are only touching the treetops today. We have limited time, but we wanted to first illustrate what scale looks like in practice, because I know we've said a few times it's a large scale implementation. We're currently holding approximately 2.7 terabytes of multi-source real-world data in the OMOP schema that we have at Kythera Labs. This is a massive amount of data. When we make changes to the OMOP transformations to parts of the ETL, we can refresh that schema in approximately 60 to 90 minutes. And the reason I'm mentioning this benchmark here is because having that flexibility to iterate, improve, make changes very quickly has been a massive accelerator for our organization. And it has allowed us to implement the OMOP standard in just a few months on such large, complex data sets. This is also a cost-effective process from a compute perspective. We don't want to be breaking the bank every time we have to refresh our OMOP data set. So again, we want to be very smart with the technology that's underneath the OMOP implementation. So here, one of the takeaways that I often think about is this story isn't just about building a large ETL. It's about building a pipeline and an infrastructure that's very efficient and reusable. And why do we care to do that? Because we're ultimately thinking about the downstream uses. One, we want to be able to improve, iterate, refresh the OMOP output as frequently as we need. Two, we want to be able to make sure that we can bring over some of those familiar tools and start to provide an experience such that if you want a data quality dashboard, CDM onboarding, familiar community tools, you can have that on top of such a large database. So you can really find what is it, what slices of the data would you take for your specific research question. And last but not least, building the foundation for more federated research use cases, having transformations and technology that are easily shareable and auditable is one of the stepping stones in that direction for us. We can go to the next slide. Perfect. Thanks, Liam. Here, we just wanted to spend a minute connecting back with where we started with Liam's presentation, just zooming in on one particular tool, the data quality dashboard. As Liam mentioned, the open source tools, in this case, we're highlighting one that works directly on your OMOP schema, so that's post your transformations. These are R-based tools, and they're very, very good for small to medium data sets. We found that at scale with very large amounts of data, performance breaks down. And what we also experience is that an R-based tool like that doesn't run out of the box in our cloud database, Databricks, AWS, Salesforce, whatever you're on. It wouldn't run natively. And what we didn't want to allow for is users pulling copies of the data and running these tools on their local machines. For us, that was a huge governance risk. So we took an interesting approach, bringing those tools over to our platform. First, we containerized a select handful of them. We focus on Data Quality Dashboard and Achilles as a first pass. And then when we run those tools, we actually do something that is not on the slides, but I think is really important to highlight. We separate the workflow for these tools in two functional parts. So think about part one, which is your query generation. What do we mean by that? A package like Data Quality Dashboard is essentially a big collection of predefined SQL queries. They check plausibility, conformance, completeness. Those are the mains that Liam highlighted on your OMOP schema. These queries are the logic for the check. They don't need to run in R by themselves. what we do need to make sure is that those are executed against the OMOP database. And same thing with second part, query execution. Traditionally, when you're running these packages in an R-based environment, both the query generation and execution are handled locally. And that can become a bottle scale when your OMOP schema is very large, holding a lot of data. So by decoupling these steps, we can use the SQL warehouse on Wayfinder and execute the queries directly on our OMOP schema very fast. So the benefit here from a community standpoint is that we're using the exact same package, the exact same logic. We're not changing that. But instead of running everything inside R, we're containerizing the packages and we're running them natively on the cloud against our OMOP database. So what we experienced with this setup is that on the approximately 2.7 terabytes of data, we can run data quality dashboard in 20 to 40 minutes. In the beginning, before we teased apart the workflow for these tools, it would take us 16 plus hours to run these tools. We would have to leave them overnight, right? Not sustainable. So big picture here is this collective strength of the open source tools, being able to always use them on your OMOP schema, but also being very clever with how you have set up your technical foundation so that you can run them efficiently at scale. And then last but not least, this is a modular solution. So we'll be adding more tools as our needs evolve. And that's pretty easy to do. So it's exciting to see that first tools enablement on platform. And next slide. We wanted to show you just a few descriptives of the OMOP schema at a glance. Again, we're just touching the treetops today. We have brought over 580 million patient records. Keep in mind that we're not talking about unique patients. We're in the process of developing an algorithmic solution to make sure that when patients from different US geographies come to us via EHR claims, other data sources, we can reliably identify if we have the same person in the database. But what's important to highlight here is the semantic coverage of the OMOP dataset. It's quite impressive. We have over 84,000 unique condition concepts. So pretty broad clinical phenotypes. So we're very excited for that because that is what will enable us to start interrogating the data and figuring out what fit for purpose use cases we can support. There's, of course, a lot more descriptives of the data that we can provide, but we just wanted to show you here the big highlights. And again, the key takeaway isn't just about the volume of the data, it's about the technical efficiency with which we've been able to create this OMOP schema in just a few months in a cloud native environment. And we can call on the open source familiar tools that we want to use as OMOP practitioners. So to recap, here's a summary of what we've built. We have implemented a multi-source OMOP pipeline across large-scale claims, EHR lab sources, in collaboration with the Hive. Their expertise, technical and subject matter has been instrumental to accelerate our OMOP journey. We have run transformations and QI checks natively in Wayfinder with the familiar Odyssey tools. And we've learned a few things along the way. We've known and we wanted to build a very modular, reproducible solution because we have improvements to make, we have work to do moving forward. But we've really benefited from that staging of EHR and claims data and that initial refinement of the data before mapping it over to OMOP, because now that we're independently managing our OMOP schema, we can trace steps back, we can modify transformations, we can independently make improvements in the long term. We also found for teams, technical teams who may be new to the OMOP standard, of course, there's a little bit of build-up time, right? It takes a little bit of time to learn your way around OMOP. It's a relational model and how to make the best sense of it. And then last but not least, we keep mentioning the data quality tools. Those are very important. Running the tools as the community knows them without making any changes but running them fast and efficiently in a cloud-based environment has been really important, thinking about growth and scale. So let me just give you a couple nuggets on what is next on our OMOP journey. We're continuing to improve what we have. We're currently working to set up automated refresh and updates of our OMOP schema so that we can have that on a set cadence. We're working to deploy, I mentioned to you, the solution to programmatically identify which patients may be the same or different across various real-world data sources. We're moving forward towards AI-enabled cohorting on OMOP formatted data. Ideally, we want to move past having to know which code sets, concept sets are relevant for particular research questions. This is where we see a really big opportunity to streamline identifying those with AI. And last but not least, we're always on the lookout to add additional data domains to our OMOP implementation. Next in line is adding additional prescription claims data into the OMOP schema. So that's it from me. Thank you so much. Looking forward to any questions and a discussion with the team.
Thank you, Alex, and Liam, for that very insightful talk. So if anyone has questions, please send them in the chat. Meanwhile, I see the poll results, and I see that the majority of people do use data quality checks as part of data harmonization process. So that's really good to hear. And the majority of people do use Odyssey tools. So now I'd be curious to know what other tools people are using. So please do inform us in the chat. We'd be curious to know. So since a lot of people do use Odyssey tools, perhaps it's nice to get into more discussion around them. For example, both in Liam's presentation and in Alex's, there was a result panel from Data Quality Dashboard, and which shows the pass rate of all of these tests. So I saw 70 something and 90 something. Liam, perhaps my question for you would be, is there like a gold standard, like p-value that you take, or is it complicated?
Good question. So I know in theory, everybody always likes to get the highest test scores. And of course, getting closest to 100% is always nice, but it's also good to understand. It might sound a bit harsh to say the limitations of your data, right? But if you are not mapping your measurements, as an example, to the absolute scrutinous level of detail that you can, right? If you're not mapping every single unit and every single possible value and everything, or you might be mapping the more general concept, then some checks may fail that you may not be able to pass in general, right? You may have, or a good analogy is drug mapping. Sometimes people have to map drugs to a higher level that's not deemed so standard, but it's the best way to represent their data. And this may still be a good use of it, but from DQD's perspective, it might go, hey, 40% of the drugs that you've assigned aren't standard according to our most up-to-date vocabulary. And in some cases, you might be able to change this. In some cases, you might not. And it's also about understanding this. So also, when you've got data at such a large scale, it's almost impossible that you're going to be able to map everything, right? So that some data quality checks will always fail to a certain extent. And this is just about you knowing the nuances of your source data and what's important for you to focus on. Right?
Alex, do you agree with that?
Yeah, I think this is actually a really, really good question and topic as a day. And I'm thinking back to a few months ago when we launched the data quality dashboard, we ran it for the first time, right? And you have thousands of checks. So when you're a new user of these tools, it can be overwhelming to know exactly what information is really important. And I remember we had this discussion on prioritization, right? Knowing where to look for which checks and knowing what to trace back so that if you are to iterate over your mappings, you're getting the most return, right? And I remember we spent quite a bit of time with Liam and team walking through what is most important from these checks and how does that translate to updating the transformations, the mapping codes, and then rerunning to get the updates, see if we've made progress. And I felt like guidance in knowing what to prioritize and what to modify first for us was really important, really beneficial.
Yeah.
Because it's easy, you can get lost in wanting to fix every little part of your ETL, or maybe trace every element from your source all the way to OMOP, which you can do and you should do. But when you're wanting to move fast and make the most out of OMOP, you got to know where to place your bets, right? So what is it that is absolutely a must improve, right? You cannot see an entry for a particular patient if you have a death listed, things like that. So this is what we focused on as a team. I thought it was really beneficial to approach it that way.
No, that's a really good point. Also, just in reference to that, like one of my examples was, oh, do any events occur after your death, right? But that's an exact example. Maybe a hospital might record your death date, and then they might record your cause of death as a disease, and that might only get stopped in the record system a month after your death, right? So from an engineering perspective, or the data holder, that makes sense to you that the condition might only end after the patient dies. So again, DQD might go, hey, that's a bit weird, but you can just say, it's okay, don't worry, we understand that, and we understand that that's just an aspect of our data that's there. And it's not necessarily evil or bad, or it doesn't decrease the quality of your data. It's just, again, something to understand. I think that's really good.
Yeah, and I thought, Liam, I thought it was also interesting how with some of these tools and checks, what becomes illuminated for you in some cases is situations, patterns in your data that you may not have thought about right off the bat. What do I mean by that? I remember with some of the prescription data, we ran into instances where you would have a patient on a particular medication, and then we didn't really know for how long there was missingness in the data. So you don't know for how long the patient is on that drug. For some conditions, that's particularly important. And these are some of the patterns in the data that we were able to shine light on and then discuss, well, what decision do we make moving forward? How do we implement? Do we want to do something like a 30-day rule after onset? For onset offset of drug use, and other examples like that, where I thought, wow, we wouldn't have seen this otherwise.
Exactly. Yeah. And I think you'd be surprised, right, by how many hospitals and clinics, as well as companies and organizations, are utilizing such rules, right? Where they'll be like, okay, we know that we cannot trust our EHR to be the most perfect, meticulous system, so we're going to do an assumption that if a patient has this drug, they had it for at least 30 days, or a 60-day, 90-day mark, or they might align it to a disease occurrence. But that's really good, right? And that's where us at the Hive, we really often then work close with the people who own the data or who know a bit more about the data, right? Because we have to interrogate it, but that also means we have to interrogate somebody else who knows about the data, right? It's not as easy as looking at a white rabbit scan report and being like, okay, now I know everything about your drug data. There's often questions that have to be asked. Sure.
Actually, on one of the topics, namely from source to OMOP. Perhaps Tim, would you like to give a little bit more information about rabbits in a hat and how that looks like?
That's a nice
Well, if there are any questions, please let us know.
So this is when you open up your rabbit in a hat software, let's say, right? And you, on the first image here, you see this can be considered your database or your data table, right? This strangely titled document or CSV file. And this is inherently a white rabbit scan output, right? Which looks like this. So this is what we will use, or the Odyssey community very often utilizes to profile your source data. It's a Java tool that will sort of look into your source data without us having to get our hands dirty with your source data and have at least some of that privacy emphasis. And what this will do will give you a nice output on what every single value contains, for example, given that the field is not doesn't have privacy issues on terms of the occurrence of the values. And then us as honesty experts, we will then make the decisions on what to manually map, right? So we know, okay, this is one concatenated data set which has multiple tables from hospital events into one. So therefore, we know this table is going to be mapping to the person, measurement, condition, drug, visit tables of the OMOP tables, because all of this information is there. And then we know, as an example, for the person table specifically, as you can see on the right then, which is an example of how we might map demographics. And this is what I meant by manually mapping. Each one of those arrows you see one of us would have drawn, or with whoever owns the data, whether that be a research institute or a company. And we will always confirm these, right, with the people who own the data to make sure that they agree. Right now, it's a bit of an easy analogy with gender going to gender content ID and ethnicity going to the race or ethnicity concept, or age going to year of birth. These don't seem so trivial. But once you start having, like Alex mentioned earlier, with drug tables that are a little bit more nuanced, and it's not so clear when a drug was taken or given, or was it dispensed, or was it just recorded? And all of these questions are very valid, but it's not always so easy to answer. And this is what I meant with this sort of initial exploring your data, but also it's in a way its own data quality check, right? Because if I know that subject ethnicity is empty, 99% of the people, and that it's not so relevant or useful to map, then this would be something to also incorporate here in the documentation. And the nice thing about Rabbit in a Hat, it will take all these mappings that you've then done, and then it'll give you documentation of to a certain extent, a very choice, as well as it can generate you the test framework, which we found to be very useful when sort of testing your ETLs as well. Yeah, if that sort of encapsulates your question.
Thank you. So there's also a question, well, sort of related from Chris. How do you go about checking OMOP map data to source data to see if similar or expected results are seen? Is this something DQD does, or do you need other tools?
Let me go make sure I understand the question. I'm not sure if I understand the question. So the question is how you go about checking if it's mapped as expected or so.
I would interpret it that way, that the OMOP is doing what we expected, given the source data.
Yeah, so that's something that DQD will do, right? DQD has all these automated checks so that it can identify. So for example, if you're accidentally making duplicate people, that's a big no-no in the OMOP world, right? You need to represent each person as a unique person. No matter how many diseases, surgeries, and procedures they have, it's still one person record. And that's what DQD will really enforce you to do especially. And then we go on to more of the plausibility checks, right? If you have 50,000 people who underwent a cesarean section and 20,000 of them are biologically male, you know that something might have gone wrong with either your source data or your transformation process. You know, if out of the 100,000 people with uterine cancer, 30% of them are classified as biological males, again, you know that something odd has happened, right? And this, obviously, these checks then are then more relevant to gender-specific or sex-specific norms, but it also goes to measurements, right? If your blood pressure measurements are in the thousands, then DQD has that little bit of that domain knowledge to say, that's a bit strange. You should check that out, you know? And again, sometimes these plausibility checks can be explained by decisions you make as collaborators on transforming your OMOP. But still a very good question.
Alex, did you want to elaborate there?
I think that was a really, really nice recap from Liam. The only thing I would add is what we experienced again which was useful when you get that information, that signal from DQG. Hey, you should go explore. It doesn't quite make sense from going from source to OMOP. Then of course you do have the freedom to do separate analyses, right? You're not limited to what you see in DQD. You can interrogate both your OMOP schema, trace back to your source, and dive a little bit deeper to confirm certain assumptions. So the DQD can, again, kind of point you in the right direction, which can be really useful when you have a lot of things that you may want to check in the data.
That's a really good point. Me personally, as a data engineer who works with OMOP, I would always push to the use of source values and making sure, okay, we see that the patient had a term indicating cancer. Do they actually have a cancer code, right? And it's sort of these little simple things that really will also be additional fantastic steps as well. Yeah, sure.
So if we zoom out a little bit from the tools, so Alex, you of course mentioned all the steps you're taking for scalability and improving operations, etc. So how has adopting OMOP helped you in your operations?
Yeah, sure. It's something that we think about quite a bit, and yeah, we can actually showcase a super simple example. For those of you who have been using OMOP schema for a long time, you would say, wow, that's easy, right? But that's the point. So having EHR claims, prescription data all the way into one singular place in our OMOP schema has made things operationally much easier for us in terms of how we query and how we analyze data internally. And one big benefit that I have experienced is being able to pull related concepts, synonyms, children of concepts in a very efficient way. So what do I mean by that? Instead of traversing several different sources to find patients with obesity, I can go to OMOP schema, I can write a simple query, and I can quickly get all the patients who have a certain condition or a related condition in just a handful of minutes. That saves a lot of time and eliminates a lot of data wrangling. And there's one more point here that we think about, which is having the OMOP schema paves the way really nicely for us to bring over some AI tools and automate some of these steps, right? Ideally, we may not even want to write SQL queries. We may want to just describe what specific patient characteristics we care for. And there are some community tools that allow you to do that in a more programmatic way. But for us, we're looking ahead and we want to leverage the OMOP schema in a new way from a technological perspective so that we can keep improving on how we can access the real world data assets that we have in-house and make things easier, faster, simpler for us, more accurate, right? Pull all the related code concepts automatically. Don't rely on a human to know which ones are relevant. So those are some of the immediate benefits we've experienced since transitioning over to OMOP.
Okay. Thank you, Alex. We have 2 minutes left, so I'm just gonna kind of give you a minute to tell us about, your sort of usage of AI and what benefits and pitfalls you've seen. More of a list than elaborate explanation because we don't have time, but we'd love to hear a little bit about your experiences.
Sorry, Azadeh, was that an audience question or for me or Liam?
I just thought to ask because AI is very much interesting for everyone. So I saw that you've been working with it a little bit. So I thought if you could share with us some of the benefits or pitfalls that you've seen.
Oh, sure. Yeah. And I can go really quickly. Yeah. So one of the biggest benefits that I think we should be thinking about, and I've seen quite a bit of discussion in the Odyssey communities on that topic as well, is automated recognition of codes, right, and code sets. So we have those coding schemas in OMOP format as well, SNOMED, LOINC, others. And it can be difficult for an individual user, human, unless an expert in a specific disease area, to know exactly which elements are relevant to persist in your cohort for your specific study questions. And so automating some of that, we have the right tools, we have the right models to be able to do that, so that, again, the process is less error prone. We want to accurately pull in all the relevant codes, all the relevant concepts, without going back and forth thinking about, did person A or person B did that correctly, verifying across people. So that's just one example of a problem we're thinking about.
Thank you, Alex and Liam. And thank you to all the audience who sat through this. Please give us your feedback about this session. We'd highly appreciate it. And don't forget to register for our next webinar all about FHIR and OMOP on 23rd of October. Thank you very much and have a good day.
Thank you.
Thank you, everyone. Thank you, Alex.